But can you see now?
Yeah, I can.
Okay, then I will continue.
Mm.
So yeah, there is a two different way for double stand break to. Uh, uh, there's two different way to DNA damage. One is direct effect, and the other one is indirect effect, which mean single standard break or double standard break. Also, in the double standard break, there is the uh, uh, clustered double standard break and non-clustered double standard break. And when the break happens, reactive oxygen species are there to repair the damaged DNAs, and they go into two different pathways. If it's low LET or high LET, if it's clustered damage or non-clustered damage, which means two of the lesions are damaged in very different path. But we are interested more in the double standard break than the single standard break, and we want to find out, or I want to learn how this double standard break repair, which pathway the DNA will take when there is a high LET and low LET, because low LET can cause double stand break as well, but simple double stand break. High LET will do complex double stand break, which is cluster DNA damage, and which path DNA will take to repair. So for the cell repair, once double stand break happens, it will go through three case pathway: non-homogeneous end joining, and homogeneous recombinations, and alternative end joining. So for the non-homogeneous, non-homology end joining, it is an error-prone repair pathway that function through the cell cycles. So there is two QU7s. They will connect together to in the cell cycle sometimes to repair the damage part of the DNA, get them connected together, then it will function. The other one is the homologous recombination, and it's an error repair pathway that require intact homologic DNA as a repair template and is active in the later S and G2 phase. So it will need replication for the code in the S or the G and the GS phase. The last one is the alternative repair DNA for double stand break. Is initiated by end resection. This one, not too much wakarimashita. Wakarimasen. Hi. But the paper introduction, it's titled as Assessing the Relative Contribution of Double Standard Break Repair Proteins as a Function of LET. The aim of this paper to evaluate the relative importance of various double stand break repairs pathways in normal cells following low and high LAT exposure. By combining experimental data for 6 different LAT using 4 different radiation types, with alpha radiation, X-ray radiation, proton, and carbon. X-ray protons are used for low LET and intermediate LET. Carbon ion is high LET, and then alpha particle for very high LET. So the methodology, they prepare appropriate cell concentrations for seeding. Then they seed the cells, RPE1 cells, nick out key DNA repair genes, nick them out, and then they get irradiated by the 6 different radiation types. Then after the irradiation, clonogenius assay used to determine the radiosensitivities and the kinetic of the DNA double-strand break repairs by using a marker 53 BBI foci. And after that, they used something called MIDARAS, which is a mathematical model designed to predict how the cell responds to radiation by simulate DNA damage, simulate the fundamental physics, and by the biological process that lead to cell death to determine the cell fate and after the radiation, and they model how the DNA repair. The results of this paper showed that different LETs with different energy for 0.5 is X-ray, proton 1 kV LET, and also proton again 12 kV, then carbon 34, then carbon 73, and then alpha particle 129 kV per micrometers. So, and these figures show the order of cell sensitivity for most to least across all 6 radiation type with different LET. Most sensitive cells are those who has lack on the ATM protein or the LEG4 proteins, and the least sensitives are those who lack of B53. So the figure showed the survivor curve becomes steeper, more linear, and the shoulder of the curve shrinks, which means higher the LET, less important how sensitive are the cells. LETs will cause the damage without caring about the cell sensitivities or protein. And the pathway for repairing is non-homogeneous and joining, more importantly for survivor, even for high LET radiation, due to the sensitivities for the proteins such as the LIG4 and DNA-BEC, which they play a huge part in the non-homogeneous ending joining reparation path. Also, the homogeneous recombination is important, but less, as cell BRCA1 are more sensitive than wild type cell, but constantly less sensitive than the non-homogeneous ending joining deficiency. So the B53 proteins in the cell are the most resistant to the LETs, no matter high or low. And now the other figures shows that the double standard break repaired measured by the foci marker for each cell line after exposure to 2 gray for different qualities radiation. In here, number of remaining double stand break over time tracked by counting the marker in the cell nucleus at 1, 4, 8, and 24 hours after exposed to 2 gray of dust of radiation. And the figures show that repair kinetic slow down with increasing LET. So in these 4 figures, with increasing LET, the repairs will take longer time. In wild type cell, disappearance of the marker after 24 hours was efficient for X-ray exposure. Whereas for higher LET, take longer time. Even after 24 hours, still they did not, there is a repair is not happening. And in here we can see that fewer markers are induced at higher LET at the residual damage with LET. Initial number of 53 foci detected at 1 hour decrease as LET increase. There is a strong approximately linear increases a residual damage with increasing LET for all cell line. That one is also what I'm saying. And the DNA protein pKa cell showed that steepest slope, suggesting that the DNA pKs protein become important for processing the complex damage induced at higher LET. So also summary of those all four figures, that the first figure A correlation between RBE and LET for all different knockout cells line against the LET of the radiation. All cell line show an increase in RBE with increasing LET. Figure B is the correlation between RBE and LET fit slope and X-ray sensitivity. It's a plot of the slope of each line from A, slope for each line here, to look at how quickly RBE increase with LET against the cell. And there is a strong linear correlation that cells are more resistant to X-ray, show a steeper increase in RBE with LET. C, which is the sensor enhancement ratio, they call it SER values, for each proteins across different radiation. For all knockout proteins, the SER is somewhat lower for particle exposure than for X-ray. But the ranking of the protein importing remain the same, high impact, and they are the most critical protein for cell survival, those proteins. And DNA-BEC and BRCA1 and Artemis, you know the names. And below or no impact are those proteins FNCD and B53. And finally, the correlation, it's a correlation between the sensor enhancement ratio and the LET for particle radiation. It tests the importing of protein change with LET. There was no significant slope for any of the line, so the sensitive enhancement ratio values for particle radiations are relatively constant across different LET. Relative importing of a specific DNA repair protein does not depend on the LET of the radiation. Protein that is crucial for surviving in X-ray damage is equally crucial for surviving proton or carbon ion damage. And then discussions in this plot where there are all the 8 cell lines and all the 6 radiation equalities. It's too smooth. Increase in RBE with LET can be explained primarily by the increased special clustering of double stand break, which overwhelmed the cell standard repair way, mainly non-homogeneous ending joint, and leads to more misrepair and cell death without requiring a fundamental switch in path choices. So in another way, is RBE with LET are caused can be explained by the different clustering of double stand break. Also, the alternative ending joining rules at high LET pathway might become more prominent at repair complex damage than non-homogeneous ending joint. Uh, Protein-specific functions within the same pathway, different protein can have a distinct role, different role. Steeper increase in residual damage for this protein, DNA BK, AK, still plays a feature lower in complex damage. And finally, the historical view was that the DNA repair play a less role after high LET radiation, and that complex damage was preferentially repaired by the HR pathways. So HR pathway is the most likely to repair after high LET damage. This is the historical view. But the funding of this paper with data strongly support the view that non-HEG pathway remain the most critical for cell survival, even under high LET conditions. The loss of NHEG gene cause the greatest radiosensitizations across all of ET. This paper suggested that NHEG is the most likely pathway for high LET. And for heavy ions like alpha and carbons, it is obviously that the repair is very slow and less efficient because of the high level of resistance which for the misrepaired or unrepairable damage. Whereas for X-ray, it's faster and more efficient. Most damage is repaired correctly through the non-homogeneous ending tuning. For protons, the repair is lower than X-ray, but faster than high LET carbons. It can happen in the non-homogeneous ending joining and in the homologous recombination. And then to conclude, most patients receive standard treatment despite clear evidence that cancer of the same time can exhibit vastly different radiosensitivities. One of the crucial determinants of cancer radiosensitivity is the DNA damage signaling and repair pathway activities. The tumor genetic profile and its DNA repair capability determine its response to radiation. HEG is the most important for survival, and the P53 protein in cells are the most resistant. And with this, I come to an end. Arigatou gozaimasu.
I want to ask you some questions.
どうぞお願いします。
Question number 3.
Number 3.
そうな、どうだ。Method.
Result?
This one. This is
This
method.
one.
Yeah. This one. So, I want to know in detail about mechanistic DNA repair and survival model.
はい。
So, what can I take information using this model? I cannot follow your this data in this paper.
Okay, what we can find is from this model we can measure the radiosensitivities of the cell for this different LET, for a different dose range, relevant dose range, fit using the linear quadratic models to predict the probability of which path will the repair take, because this model can detect nick out proteins. They nick out some proteins which are the most likely to be repaired, and then it can simulate fundamental physics, simulate the DNA damage appear if it's double strand break complex or double strand break non-complex.
So are there data using this model?
Are they data?
Yeah. In this
Yes, I think they are in this paper.
Yeah.
Yeah, there is a table.
So, for example, this next. So this is a set of biological data, right?
So this, so for this, the different proteins,
Yeah, I know.
and then what they did in this one, mechanism is to find how the radiation will respond for each of this protein.
Yes, yes. So this data is all biological data using cell line, each cell line.
Hi.
They cannot use model.
Oh, yeah. But the result I am sharing is not. There are another figures I did not share here, the papers.
Oh, really?
There are other things that, for example, at the tables for this, I did not share here. I just share what I think it is interested for me. So, for example, why I share this? Because it shows what is the most sensitive and what is the least sensitive. And then why I share this? Because it's this one. It shows how fast is the repair for different LED. The result they take from this final result, I did not put in this slide. I did not share. And then because the Madeira or these things they use make them develop what they call it the sensor enhancement ratio.
We cannot take this information using that model.
Hi. We can take this information using that model, which is how sensitive each protein to make how sensitive each protein in inducing the type of radiation and the LET. But what is, and then after this, they find out that historically we know, or it was known, that DNA repair is not important for high LET. And most likely it will be repaired by the HR. But because of the funding and the media they used and this SER, sensor enhancement ratios, now they believe, as the paper suggested and the resource suggested, that even in high LET, NHEG is the pathway for repair.
Can you use this model? You know?
Equation mathematically, I understand the math, the physics, the equation, but I cannot use